Abby, Ashlyn, Lily, Laurel, and Kyla. I wanted to give y'all a video to cover some of the things that we didn't cover in the lab today that will be important for y'all to know. So if you want to get out your notebooks, it will be important for you to take notes, and there will be a lot of captions so that you know terms. Make sure you write those down.
Here's what we're going to cover in this video. We're only going to be looking at animal cells, and we'll be focusing on the cells that we looked at in lab, talking about the stains that are from the lab, talking about organelles and their functions, and also cell specialization.
So the first thing we're going to do, I went back and did some of the things in lab so that y'all could see some pictures. One of the first pictures that you're going to see will be a picture of the dry mouth cheek cells.
Notice the difficulty you may have finding the cheek cells. The black pointer is pointing to a cheek cell, and then there's also a couple to the left of the pointer. This is a dry mount. I want you to compare this to our next image, the wet mount.
The next picture that you will see will be the wet mount cheek cells.
Notice that it's already easier to see the cheek cells in this wet mount. The reason for that is the light shining through the base of the microscope. When it hits the water, kind of like that air bubble you see on the far right side, the outlines are more crisp, so your cells you can see better. You can probably see the cell membrane, the cytoplasm is a little bit darker, and there's also the nucleus, which is a little bit darker as well, and relatively small compared to the rest of the cell.
This image is of the wet mount cheek cells that have been stained with the iodine gram stain.
Iodine stains stain carbohydrates, specifically starches, which is a carbohydrate found only in plant cells. When you're staining plant cells that have starches in them, the starch will actually turn the iodine stain black. Here, since we only have an animal cell, the iodine stain stays yellow and shows you contrast. As you can see, there's more contrast between this cell with the iodine stain versus a wet mount with no stain. Here's what the cheek cells would look like being stained with methylene blue. Here is our cheek cell after it's been stained with methyl blue. The nucleus is the most obvious component of the cell because methyl blue is most attracted to the components that make up a nucleus, which is the DNA and the RNA. You'll also see the bacteria is labeled here because methyl blue is also attracted to bacteria. It's easy to see here in the cell. Do not try and label bacteria in your cheek cells that y'all drew. We didn't use the methyl blue stain, so it's not very easy to distinguish bacteria from other things that are in your cell. But it makes sense to find bacteria on your slides because when collecting cheek cells, you're definitely going to be collecting other things in your mouth. Bacteria is one of those things there's a lot of in your mouth.
This picture is of my red blood cells.
Okay, not a lot of detail. I'm going to go through actually the correct way to create a blood smear slide. So with Kyla's example, there was so much blood that it was really hard to tell one cell from another. The way that you do that is you take another slide, angle it 45 degrees, and rake it across the slide with the blood. It looks like you're raking off all of the liquid, but once you've done that, you look down and you have a smear of blood. That smear of blood is one cell thick. So when you put it on your microscope, you're not going to see a pile of cells on top of each other. You'll be able to see one from the other.
This image is of the red blood cells that have been stained with Wright stain.
So here are my blood cells after being stained with Wright stain. It's not obvious, but where the arrows are pointing indicates where the dye shows a different type of blood cell, the white blood cell or leukocytes. I'm going to show you a better image so you can see what the dye does to differentiate blood cell types. Here is where leukocytes are dyed with the Wright stain, They are very bright purple and can be distinguished easily from the normal red blood cells around them. So here is how all of the preparation for making the slide with the right stain and the blood happened. First, I put the blood on the slide, raked another slide across my slide to get the blood smear, let it dry. Then I added the right stain, let that dry. Then I took both the blood smear and the right stain and the slide and I put it in a bath where I gently rinsed to take any excess right stain off. This image shows you just an empty slide in the bath, but originally I put the whole slide in there with the blood and the right stain. Then I let that dry and put it under the microscope to take the pictures that you just saw. Gram stains, like our iodine gram stains, are performed on body fluid or biopsy when infection is suspected. Gram stains yield results much more quickly than culturing, and it's especially important when infections would make an important difference in the patient's treatment and prognosis. This is really cool. Methylene blue is used in aquaculture by tropical fish hobbyists as a bacterial and fungal infection preventative on the freshwater fish eggs. It's also used commonly as an additive in solution in which infected or newly purchased fish are immersed. When the fish are dipped in the methylene blue, it serves to fight and kill off offending organisms, as well as it increases the oxygen carrying capacity of the fish's hemoglobin, which means they can breathe better in the water. Here you're seeing a doctor preparing a blood smear with the right stain. Right stain, because it distinguishes easily between blood cells, is widely used for performing differential white blood cell counts, which are routinely ordered when infections are suspected. Also, the stain can be used for the chromosomes to facilitate diagnosis of syndromes and diseases. This is exciting. I like this part because we're going to be talking about cells and their organelles. The pictures that y'all have drawn with the cells that you looked at in lab, there's only going to be a few things that you're going to want to label because those are the only ones that you know for sure when you're looking at your cell, so I'll make those clear. But I want y'all to also know these terms coming into class, and we'll actually talk about a little bit more organelles in class. Here we have a typical animal cell. We'll look at each of the organelles within the typical animal cell, and an organelle is an individual functional unit of the cell, or another definition of an organelle is any of a number of organized or specialized structures within a living cell. As y'all noticed in lab, every cell you had a cell membrane. That's the layer that protects the cell. Then it also, you noticed, had a nucleus, and I drew my nucleus here in purple. And the reason that I have two circles around the nucleus is that the nuclear membrane is different than the cell membrane, is that it is a double lipid bilayer. So the cell membrane is only a single lipid bilayer, and then the nuclear membrane is a double lipid bilayer. So the nucleus has its own nuclear membrane, and then in the center where the genetic information stays is called the nucleolus. Y'all were able to see on maybe some of the cheek cells a nucleolus, maybe not. If you don't see it, don't label it, just label the nucleus. You all know what cytoplasm is, and that's anything within the cell membrane and outside of the nucleus. So it includes the organelles, includes the ribosome, it includes the water, it includes the molecules that are dissolved in the cytoplasm, and something that we'll talk about later called the cytoskeleton that gives the cell its structure. Also, I have in blue, and the dots all in the cell are something called ribosomes. You may see something like that in some of your drawings, but don't label it. We don't know for sure if that maybe is a bacteria or paper towel, but ribosomes are in typical animal cells,
And then a few of you may have seen a vacuole in your cell. Vacuoles are just storage organelles. Things go there maybe to be excreted from the cell. Those look like small air pockets within an animal cell. Within plant cells, they actually can be very large.
Lastly, we will look at the mitochondria. Mitochondria, or organelles, called the powerhouse of the cell. This is where most of the ATP is made, which is the energy for the cell. Mitochondria, as similar to the nucleus, has a double membrane, and interestingly, this is the only organelle that also has its own DNA. In genetics, mothers pass down their mitochondria to the children. You only have your mother's mitochondria. The double membrane and the separate DNA supports a theory that mitochondria used to be their own separate single-celled organisms and were at some point engulfed by cells, maybe to be eaten. And because they gave an important role to the cell of creating the energy in this way, they were kept in the cells, and they have remained in the cells since.
Now y'all have kind of an idea of the variations just within animal cells. So when we were looking at cheek cells, they looked kind of squarish. When we draw a typical animal cell, it's this nice round cell. Your paramecium was sort of bullet shaped, and the amoeba cell was kind of like a splatter of paint that I told Kyla. I don't know if y'all all heard that analogy. They look kind of irregular. So we'll look at what maybe makes it important for the animal cells to have different shapes. So this is specialization of cells. Our paramecium has a bullet shape, and what we didn't see under the microscope is that paramecium are covered with small hairs called cilia. They're very small, so you can see already a specialization for this cell compared to our amoeba. The small cilia are for transportation. Now, they do not look like they could hold the cell's weight, which is very accurate. So this cell is found in some sort of water environment, typically. Uses the cilia to be able to travel within the water. If it comes into contact with an object, then it reverses the movement of the cilia, keeps going forwards and backwards until it gets around that object. Very low productivity of movement, but works for the paramecium. You'll see that I also drew this, which is called an oral groove for a paramecium. The paramecium is covered in these cilia, so the way they move is if you imagine a field that's being blown by the wind, how the grass moves, that's how the cilia move on the paramecium. This oral groove, the cilia will actually pass food into the mouth of the paramecium. And again, all of this works because the paramecium is within the environment that's water. The bullet shape helps it move throughout water. It's going to be more streamlined that way. And then the cilia are not overdeveloped appendages by any means, so they simply just propel the paramecium forward. So the amoeba, it was the second cell that we looked at, a single-celled organism. We saw the nucleus, and this cell does not have any cilia for transport. The reason that that is not the case is the transport for the amoebas is much different than the paramecium. So if amoeba saw a food source over here, it's able to push its cytoplasm into different parts of the cell, and it would push its cytoplasm into this part of the cell, slowly pushing it towards the food and maybe removing the cytoplasm there, pushing it even more, and it's going to start engulfing the food source until finally it is engulfed the food source, and you see that is the creation of a vacuole. Where now it can come into the cell and provide it with energy. So the amoebas push out into what's called pseudopods, which are fake limbs, and then they kind of push themselves around to move in their environment. So that's the reason that this cell is shaped the way that it is. One of the most common well-known amoebas is slime mold, and slime mold will actually aggregate all of the single-celled amoebas into one, and it will turn into kind of like the slug that has movement all of its own. It'll move to where the environment's right, and then it will mature and create spores that then turn into the single-celled amoebas. So it's a life cycle for the amoeba.